Hello guys, welcome to my channel. So today we'll study about chromosomes. As if you have seen the previous videos, we studied about the structure of DNA, and then after we studied about the cell cycle and the cell cycle control system. Now after learning about the cell cycle and the cell cycle control system, we can go through the chromosomes. Alright, so in eukaryotic cells, very long double-stranded DNA molecules are packaged into chromosomes. Right? So all the long-stranded DNA are packaged into chromosomes. So the complex task of packaging DNA is accomplished by a specialized protein. So there is a specialized protein that do the task to package that DNA into the chromosome that bind also the DNA and that fold the DNA, generating a series of coils and loops that provide increasing higher level of organization and prevent the DNA from becoming a tangled and unmanageable mess. Right? So in eukaryotes such as ourselves, DNA in the nucleus is distributed among a set of different chromosomes. So what the chromosome contains? So chromosome contains first the DNA. So the DNA in the human nucleus, for example, contains approximately 3.2 times 10 raised to 9 nucleotides. So the DNA of human nucleus contains 3.2 times 10 raised to 9 nucleotides sequence. So this 3.2 10 raised to 9 nucleotides are in the DNA and they are parceled out into these are parceled out into 23 or 24 chromosomes. All right. So each chromosome consists of a single enormously long linear DNA molecule, right, associated with the proteins that fold and pack the fine thread of the DNA. So what the chromosome contains? So the chromosomes consist of DNA, right, and the protein protein that fold bind and fold the DNA. Oops. And fold the DNA. So the chromosome has these two things, DNA and the protein that is responsible to fold and package the DNA very well into the chromosome, right? Because the chromosome is a really compact structure, so that's why the DNA needs a specialized protein to do that task, right? So the complex of DNA and the protein, so this together, the complex of DNA and the specialized protein, right? And the protein is called chromatin. So this together is called chromatin. So when you hear the word chromatin, you will know. So the chromatin is it has a complex of DNA and the protein that is responsible for binding and folding the DNA. So in addition to this protein involved in packaging the DNA, chromosomes are also associated with many other proteins involved like in DNA replication, DNA repair or gene expression. So there are other kinds of protein as well. So with the exception of a germ cells, that is the sperms and eggs cells, highly specialized, and also there are highly specialized cell that lack DNA. So germ cell does not have DNA, egg cell does not have DNA, such as in this highly specialized cell such as the mature red blood cell. All right, so they lack DNA. Human cells each contains two copy every human cell contains two copies of each chromosome, one inherited from mother and one from the father. So the maternal and the paternal chromosome of a pair, so those pair are called homologous chromosome. Homologs, also known as, let's write it down. So and so the maternal and the paternal chromosomes of a pair is called homologous chromosome, or also known as homologs. So this means X from the maternal and X also from the paternal. So they both have same X chromosomes. So that is, so I will just write it down here like X from both the parents, right? So the only non-homologous chromosome, the only homologous chromosome, so the only non-homologous chromosome pairs are the, okay, right here are the sex chromosome, right? Sex chromosomes in males. So the non-homologous only pair is the sex chromosomes in the male, where a Y chromosome is inherited. Let me just write it down like this. So the Y chromosome is inherited from the father, and we'll make it big. From mother, right? So Y chromosome from father and X chromosome from mother. So those are the sex chromosome and those are non-homologous chromosomes. So the female inherits X chromosome from each parent and have no Y chromosome. There are homologous chromosomes, right? So homologous and non-homologous chromosome, right? So if you don't know, so there is different sizes of, so all the 23 chromosome has different sizes. So the different human chromosome can be distinguished from one another by a variety of techniques. So there are different kind of techniques to distinguish from one chromosome from another. So like there's DNA hybridization, right? So what in that technique, it's just they color the chromosome using the sets of this chromosome, some specific DNA molecules coupled to different fluorescent dyes, right? So they dye it, and like every chromosome will have a different color. And so like an ordered display of full set of 46 human chromosome is called the human karyotypes. So if a parts of a chromosomes are lost or switched between the chromosomes, these changes can be detected. So the cytogenetics analyzed karyotypes to detect chromosomal abnormalities. So they will know, like for example, if chromosome number 12 has something like this, maybe I will just draw. So this is chromosome number 12, and let me use different color. And for example, it has this, right? So this is 12, and for example, this is 14. So you can see this 12 has been mutated with the 14, and this is some chromosomal abnormality. So just for the example, all right, right. So we saw what the chromosome consists of. Now we'll see the function, right? The function of chromosome. So the most important function is important function of chromosome is to, right, I will write it down, of chromosome is to carry the genes, right? So the most important function of the chromosome is to carry the genes. And what is the gene? The functional unit of genes is a functional unit of heredity. Right? Heredity. Right? So the most important function of chromosome is to carry the genes. And what is gene? If you don't know what is gene, so gene is actually defined as a segment of, this is defined as a, it is a, like a segment of DNA. This is not entire DNA, but it's a segment of DNA that contains the instruction. So that segment of DNA contains the instruction for making a particular protein, right? Particular protein, or it can be a RNA molecule, right? So this is the definition of gene, right? So gene has a segment of DNA that contains the instruction for making a particular protein or an RNA molecule. And most of the RNA molecules encoded by genes are subsequently used to produce the protein, right? So the gene contains instruction, for example, to create an RNA molecule. An RNA molecule in turn is used to encode or used to produce proteins, right? So in some cases, however, the RNA molecule is itself the final product, right? So most of the time, gene contains a segment, so the segment produces RNA, and then RNA produces the protein because it has how to make the protein. But sometimes the RNA is itself a final product, right? Like a protein. So this RNA molecule have diverse functions in the cell, including structural, catalytic, or in gene regulatory roles, right? So it has different kinds of function. So together, the total genetic information carried by all the chromosomes in a cell or organism constitutes its genome, right? So the total genetic information carried by all the chromosome, right? All the 23 chromosomes in a cell, all an organism constitutes its genome. So that is called a genome, right? And just for the knowledge, in addition to the genes and the specific nucleotide sequences required for normal gene expression, a large excess of interspersed DNA is also present, and this extra DNA is sometimes called chunk DNA, but that is of no use. Just for the knowledge. All right, and all we know that humans have total 46 chromosomes, 23 pairs, right? All right, so we saw the introduction for the chromosomes, right? So what are chromosomes and the function, and what is genes and what are genomes, right? So the gene is the segment of DNA and the genome is the entire genetic information. So now we'll see the structure of chromosome. So why we studied the cell cycle before? Because during the interphase of the cell cycle, right, we discussed about the different phases of cell cycle, and during the interphase, the chromosomes are extended as long thin tangled threads of DNA in the nucleus. So, as also it is referred as an interface chromosome. So, on the chromosome, there is a specialized DNA sequence found in all eukaryotes and that sequence ensure that the DNA replication occurs efficiently. So, I will show you by the diagram how it is. It looks sorry. So, maybe I'll draw it here. I will try to use a different color. Uh, Maybe if it works. More. Alright. Maybe I will make this different. So I will explain to you later that what are this different that I'm making. Maybe I will make a big of this. I don't have right now different colors, so I have just two colors, so I will show you. So the and part, so I will name it later as we'll discuss. Right, so as I mentioned that every chromosomes, the interface chromosome has a specialized DNA sequence that are found in all eukaryotes ensure that the DNA replications occur efficiently during interphase. And the one type of the one type of nucleotide sequence is known as replication origin. It's known as the replication origin. So the pink one that you see, this one. So this is our replication uh, origin. I'll write it down here. So this pink and this pink both are the replication origin nucleotide sequence. This act as a replication origin. So what it does, and there are like so many replication of origin or replication origin nucleotide sequence on this one chromosome. So one type of nucleotide, right? So replication origin. So where this replication of origin, where the replication of the DNA begins, and the eukaryotic chromosomes have contains many replication origins to ensure that the long DNA molecules are replicated rapidly. So this is the thing, the pink that you are seeing. There are I just made two, but like there are more than two replication origin nucleotide sequence on the chromosome. After that, there is another DNA sequence that forms telomeres. So this end part that you see here that I made as a block, so that is telomeres. So on the both end, telomeres. So the telomeres, so what the telomeres do? So the telomeres are at the each of the two ends of the chromosomes, and the telomeres contain the repeated nucleotide sequence that are required at the end of the chromosome to be replicated. And the main function is they also kept the end of the DNA, this end of the DNA molecules, preventing them from mistaken by cells as a broken need of repair. Why? Because so as simple as just take a shoelace, right? So if there is, this is a shoelace, you see that at the end there is something like this, right? A metal thing at the end of the shoelace. Why? It's because they put at the ends so our shoelace do not open and put in all the threads. Do not open in all the threads. So this and part is the same function as for the telomeres. So it is holding the nucleotide sequence as tightly, and so it won't spill up, and not exactly spill up, but so it is not mistaken as a broken DNA in need of repair. That's the function of telomere, right? So we have this both black end that you see, so that is telomere. This pink is our replication of origin, and eukaryotic chromosome also contain a third type of specialized DNA sequence, and that is called centromere, right? So that is called centromere, and this centromere that allows duplicated chromosomes to be separated. We studied that during the M phase, right? In the centromeres allows the duplicated chromosomes to be pulled apart, right? So during the M phase there will be kinetochore, and the mitotic spindle will attach to the kinetochore at the centromere, and then they will pull the chromosomes apart. So I will show you the process how the chromosome is separated into two during the cell cycle. So this is our one chromosome, and this is during the interphase of the chromosome. So what happened next, I will show you. Let me see how I can draw this. It's different color. Let me just make this, and then I will explain to you what it means. Right, so we have and we have our, and after that I make this it's small, but all right. So this is all happens. This everything happens in the interface, so interface of the cell cycle. All right. So first, as we see, this is our chromosome that is happening in the interface. It has our replication origin, centromere, telomeres, everything, right? So the DNA replicates in the interface beginning. So where it starts, it begins at the replication origin or origins of the replication, and proceeding by directionality. So it goes, so from this it happens like this, how it splits into two. So it happens by directionality from the origin across the chromosome. So it goes like this. So this becomes like this, and it start pulling, right, pulling outwards, right? So it start dividing from this origin of replication like this. As you can see, there are different many origin of replication, and it goes like this, and then it separates and becomes into two sister chromatids. That is it, that happens in our interphase. Then in M phase, what happens, as we know that it's maybe just a random picture that I'm making. So something like this, and maybe it has, let me use different color. Not very well picture, but something like this. Two sister chromatid, it has our centromeres, and for example, these are the mitotic spindles that will pull. So I will write it down. Portion of mitotic spindle, and this happens in the M phase, right? It separates and then cell division happens, correct? And what happens after the cell division? Again, it will be the same, but in a separate form or separate way. So this is the first one, and this is, for example, our second. Alright, so let me just draw it quick. Then our this, and maybe that is here. Alright, so these are two separate or two duplicated chromosomes, again in the interface of the separate cells. That happens, right? So this is in one cell, then it starts separating, it becomes two sister chromatid, then it is like this in the M phase, it's pulled, the mitotic spindle attached to the kinetochore at the centromere, and then it's pulled and it is duplicated chromosome in separate cell. Alright, so this is not exactly looks like, but just to explain the things that are happening in the chromosome, correct? Right, so the interface chromosome, this are a long, thin, finely thread like a chromosome, but this M phase or the mitotic chromosome is really condensed. As during this stage of the cell cycle, the DNA coils and adopting a more and more compact structure, ultimately forming a highly compacted or condensed mitotic chromosomes. So you can see the image. So this is how it looks as a mitotic chromosome, like so condensed, right? This is the mitotic chromosome as you can see on the image, right? So this is our structure of chromosome. So now, as we have seen, all eukaryotic cells, whether in interphase or mitosis, package the DNA tightly into chromosome, right? And the human chromosome, for example, that is, for example, 22 number chromosome number 22, so contains 48 million nucleotide pairs stretch out end to end in the DNA. Its DNA would extend about 1 point. So DNA would be like around 1.5 centimeter, right? Yet during this M phase mitosis, chromosome 22 measures about only 2 micrometer in length. So that is nearly 10,000 times more compact than the DNA would be if it were stretched to its full length. So this remarkable feat of compression is performed by proteins that coil and fold the DNA into higher and higher level of organization. So next we'll see which are the proteins that help to coil the DNA or to fold and package the DNA so finely. Right? Alright, so we'll see which are the proteins that are responsible for packing the DNA so finely. So the proteins that bind to the DNA to form eukaryotic chromosomes are traditionally divided into two general classes. So it is traditionally divided into two general classes. So they are histones, the first one, and the second is the non-histone chromosomal proteins. So these are the two chromosomal proteins, right? So that are traditionally that binds the DNA to form a eukaryotic chromosome, histones and non-histones chromosomal proteins. All right, so these are the two proteins, and the complex of both class of protein with the nuclear DNA is called chromatin. That you see in here. The complex of DNA and the protein is called a chromatin. So the complex of both type of protein plus the DNA with the nuclear DNA is called chromatin, right? So histones are responsible for the first and the most fundamental level of chromosome packaging, that is called the nucleosome, right? Nucleosome, right? This is a term. So this is a basic structural packaging unit of eukaryotic DNA, right? So just it's not any definition, but basic structural packaging unit of eukaryotic DNA, right? So histones are responsible. These are responsible for the first and the most fundamental level of chromatin packaging, the nucleosome, right? And this was discovered in 1974. So actually, when the scientists found this, so they break open the nuclei and then they examine with an electron microscope to see much of the chromatin. So they found a chromatin fibers with a diameter of 13 nanometers. So your screen, so the electron micrograph or the sorry electron microscope results. This is how it was seen. So the first image, that is A, you can see this. So that is a chromatin fiber or chromatin isolated directly from an interface nucleus. So it is a chromatin fiber about 30 nanometers thick. But in the B that you are seeing, so the thread-like structure, the electron microscope, it's a length of a chromatin fiber, but that has been experimentally unpacked or decondensed after isolation to show the beats on a string. Right? So an individual nucleosome core particle consists of a complex individual nucleosome core particle. So that means so the picture you have seen in the electron microscope, something like this, right? A string, and then there is like a beads on this, right? So an individual nucleosome. So these are the nucleosomes core particle that you are seeing, right? It's a DNA and it's wrapped with the nucleosome. So this individual nucleosome core particle consists of a complex of 8 histone proteins. Right, so I will write it down here for you. Individual nucleosome sorry consists of 8 histone proteins, 8 histone proteins. So which are two molecules of each of histone, right? So it is H2A. So it has two molecules of each. So H2A, H2B, H3, and H4. I will write it down. Two molecules of each. So this is how it consists of 8 histone proteins. So H2A, H2B, H3, and H4, and a stretch of a double-stranded DNA in this one nucleosome core particle. Alright, so I will show you here only. So, it's like a string wrapping around maybe. That is making sense. Anything? Right? Okay. Right. So something like this. So this. So like the string, right? It has the beads. So just a few section. You can see this is how it looks. It has a DNA, and this middle is called a linker DNA, as it's linking the other DNA. This is our this is our that I'm talking about, the core of nucleosome, right? This part is at the core of nucleosome, right? And the beads, so this bead on a string form a chromatin, right? So this is the chromatin that beads you can see on the string, all right? Linker DNA, this is the core of the nucleosome, so it contains our DNA and this 8 proteins, this one core nucleosome, right? So like for example in test tube, so the nucleosome core particle can be released, this nucleosome core particle can be released from the chromatin by the digestion of this linker DNA with a nucleus, right? So this, like in the test tube, like the linker DNA can be digested by nuclease digest, that will digest, and like maybe, or maybe I will put it on this side, right? So it will just have, after for example this is our round, right, having, yeah, but now after digestion we'll have just this, the core, right? The linker DNA is digested by the nuclease, and after that we'll just get a single round structure, right? Our core of the nucleosome. This doesn't look very good, but like this is around and like there is something wrapped around it, right? So maybe, well, if I make like this, maybe it looks like something wrapped around this, right? Okay, wait, now I got it. All right, and after this it will look like something like this. I can use a different marker. Make it. So after you will get a separate core nucleosome, right? We'll get a separate core nucleosome and the DNA around each nucleated chromosome. So that this outer part, this is the wrapping. I show you this in the pink separate thing. So this is the DNA that is around each isolated nucleosome core particle, and then this can be released. This pink section, that is our DNA, can be released, and its length determined with 147. So after it is released, then the scientists try to determine the length of the DNA. So they found it this. All right, let me remove this and write it here maybe. So H2A, H2B, H3 and H4. So after rolling is it, so we have our this core nucleosome and just a DNA, right? Or maybe not a simple thread, but maybe like this. So the length is 147 nucleotide, right? 147 nucleotide per DNA, and this is our histone, right? As histone combines with DNA, that makes the core nucleosome, right? And histone octamer, right? It has this H2A, H2B, H3 and H4 proteins, right? So this is how It is determined how they find what are at the core of the nucleosome, and each core of the nucleosome contains 147 nucleotide pair of the DNA in each fragment. All right, now I think that is enough for this video. So we'll make the another part. So in that part, we'll see how this does packaging the DNA and how this becomes like this. From this to becomes like a thread. So we'll see in the part 2. Alright guys, so that was our chromosome. We discussed the introduction, the function, the gene, the structure, how it was in the interphase, the enzyme phase, and then how it separates, and basically how it is prepared or packaged from the proteins. So we'll see in the next video how the contents, the histones and the DNA are packaged together and form the structure like this. Alright, thank you guys for watching, and don't forget